You are an enthusiastic microbiologist who's looking into a bacteria. Then as you're looking into it, you see plethora of small circular substances which are suspended in the cytoplasm. Now you wonder what they are. Now as you're looking to them, you deep dive into what the story can be, and you see they are present in many numbers. As I told you, it's a plethora. They could also be single, sometimes just one being alone suspended in the cytoplasm. Now, you could also see them in bacteria and also yeast. Now, looking at the numbers, you also sometimes think, is this essential for the bacterial cell or the yeast cell? Well, no. The sad part is it's not essential. This circular substance, which is present in the cytoplasm, which is double-stranded, replicates on its own. It is independent. The reason being, because it has its own origin of replication, anori, and it has its own genes that helps in replication, and they also have replicons, or this process itself is called as replicon. Now we wonder what these things are, and they are called as plasmids. To sometimes reduce their burden of replication or decreasing their effort of replication, what they do is they bind with the bacterial DNA, bacterial chromosomal DNA. They bind with the bacterial chromosomal DNA so as to produce a structure called as an episome. An episome. This episome will replicate along with the bacterial chromosome, thus decreasing its own effort for replication. Then as we go ahead, you think, let me eliminate these plasmids which are present in so much quantity or sometimes single within the cell. Then you do a process called as curing. What you do in this is that you eliminate the plasmids in the bacteria. Now this can happen by treating the host cell with a lot of substances. Now to remember what those substances are, remember a thermometer which is of the company T-star. And this thermometer is subjected to flash. What is this flash? They are radiations. And oh my god, it is also subjected to an acridine, that is stachrine. So what is this thermometer? When I speak about thermometer, I say it's the temperature. When I talk about T-star, I talk about thymine starvation. And when I talk about a thunder, I talk about the radiations. And then ultimately the stachrine. So these are the substances which is going to, cause the process of curing. Now I would like to classify, you as a person would also like to classify the plasmids. Now this could happen because of conjugation, and it could also be classified based on the compatibility. It could also be based on the function what it is performing, or it is also based on, that's all. These are the main categories. Like it could be because of the compatibility, or it could be because of the conjugation, or it could be because of the function, or it could be because of nothing else. Okay. When we talk about the conjugation, well, we have to know what is conjugation. Conjugation is a process in which two bacteria are, transferring their own genetic material. Conjugation is a process of bacterial transfer between two bacteria where they share genetic material by producing a sex pili. And to remember that process, we have to remember a gene, and the specific gene for that would be called as tragenes. But wait, let's get to that sex pili which is produced by tragenes. But let's talk about conjugation. So there are some who transfer their genetic material by conjugation process by producing a conjugation tube. They are called as conjugative plasmids, and there are ones which don't do it at all, like without a conjugation process. That's called as non-conjugative plasmid. Then you speak about something based on compatibility. Have you ever wondered, there are plethora of these organisms which are present within the bacteria. Then have you ever wondered why, for what reason are they coexisting together? The point is very simple. They're coexisting because they're compatible with each other. That's the same thing, right? Within a house, there are multiple people can coexist. Why? Because they're compatible with each other. So that's how. These compatible plasmids are compatible with each other. So that's why they call as compatible plasmids. And there are some other plasmids which are having no compatibility at all. They can't share the same room. Alas, that's very sad. Now, the reason why they cannot share the same room is because of they share the same kind of origin of replication, and thus it will cause competition, thus inhibiting the process itself. Now then let's talk about the function of it. Based on the function, I need to ask you to remember, we have to first remember, based on function, we'll have to remember a mnemonic called as cal met viru for, that's the question mark. Why did cal meet viru? So the mnemonic is again cal met viru. Now cal means call gene or a call plasmid. Met is the metabolic plasmid. Then you have viru. V is V for the virulence plasmid and R is the resistance plasmid. So that is a good mnemonic, I guess. Cal met viru for. So now let's talk about these resistance plasmids. Whoa, they're having resistance against what? The antibiotics. Then you have, that's why these plasmids encode for a gene which basically has a function of resistance against an antibiotic. Then you have plasmid, F plasmids. These are coded by TRA genes. These TRA genes encode for the sex pili, and that helps in formation of conjugation tube. Then you have virulence plasmids. Okay, to remember the virulence plasmids, I want you to remember a beaker which is being heated, and in that you have some heat labile and also heat stable E. coli toxins, and on the side of it you have something which is very adhering to each other. So what is it? A cup which has a heat stable toxin and a heat labile toxin. On the side of it you have adherence antigen. So what the examples for this is going to be heat stable toxin, heat labile toxin, and adherence antigens. Then ultimately you have something called as metabolic plasmids. The metabolic plasmids will help in the bacteria to make sure that it digests the things which are not favorable for it. That could be a salicylate, camphor, or toluene. Then we also know that there are some bacteria which convert urea to ammonia, and that is happening because of an enzyme called as urease, and that is again mediated by these metabolic plasmids who are coding for the gene urease. Then we also know some bacteria is fixed nitrogen, that is also because of the same. We have read a lot of these bacteria and during the process of nitrogen fixations when they are present in the roots of a lot of legumes and stuff like that. Plasmids are really ideal vectors in conditions of recombinant DNA technology because these plasmids are self-replicating and they carry things very easily, and we have already mentioned about the effortless transmission it does as an episome. So they're used extensively in recombinant DNA technology to carry the whole gene which is required for transfer. This is why we're using it in recombinant technology is again because for a selective protein production or gene therapy or such thing like that.